What's up, guys? Ben here from Velocity PT Academy. We've got a great episode here today. We've got Alex Kolliari-Turner. I've said that right, haven't I, Alex? Have I? Yep.
Yes.
Oh, awesome. On our podcast today. And what Alex is doing is a PhD student from the University of Brighton. That's correct, is it, Alex?
Correct.
Correct, yep. And he's looking into the implications of RNA sequencing in the detection of anabolic steroid use and the harnessing of the molecular mechanism of muscle memory. So it's quite a mouthful, but Alex, if you'd just like to introduce yourself, and I'll just get straight into the question after you've introduced yourself in regards to just a brief about your research as well.
Yes, so I'm a second-year PhD student at the University of Brighton in England, and I am conducting research where we are looking to recruit both current and past anabolic steroid users to further investigate the question as is there a retained benefit to someone who has taken anabolic steroids many months and years after their usage has stopped.
Right.
And we are still in the recruitment phase at the moment. And in fact, in 2019, we have one more weekend left where we will be sampling participants. That is the 26th and 27th of October. But we will be continuing to sample participants throughout 2020. We have got now over 40 people involved in this research. And at the end of October, fingers crossed, if everyone attends who has expressed an interest, then we will have even more. And this is one of the few research studies that's being conducted at the moment that is sort of coming along from various other research being conducted now over the past 15 years that is just trying to further answer this question in humans. Is there a retained benefit from steroid usage in terms of steroids potentially enhancing muscle memory, which is just the phrase to describe that if you've been exposed to steroids in the past, potentially your ability to build muscle is maybe permanently enhanced, at least with the data we currently have from animals. And the question is that the case in humans?
Mm-hmm. Yeah, no, interesting. So just for our guys that are listening to this, Alex, they might not be, as I'm not as well, clued up on some of the terminology that we're probably going to go through. So firstly, RNA sequencing, what does that mean? Just to scale it.
So DNA is the genetic material that has the blueprint for whatever the cell is. So it's the blueprint of your genetic code. Okay, so the DNA inside the cells inside your brain is the same as the DNA inside your muscle, is the same as the DNA inside your liver, and that is your genetic code. And the way that DNA is packaged in different cell types determines what is expressed. And so you could think of the genes that code for making a brain, so that section of DNA, that gene that codes for making a brain, is expressed inside a brain, but it's not expressed inside your liver. That region of the DNA is closed and shut. The same goes for the DNA and those genes that are inside that code for making muscle are open inside muscle fibers and are being expressed, but they're shut inside the brain. And so when a gene is expressed, the DNA gets converted into something called RNA, and that RNA then gets converted into a protein, and the protein is then what gives the characteristic of the cell. So inside muscle, you have, for example, the protein actin and myosin, which are the contractile proteins that cause muscle to actually physically contract. Those actin and myosin genes are open. Those regions of DNA are open inside muscle, so they will be read. They will then be converted into RNA, and that RNA will then get converted into protein, and the actin and myosin proteins are then what make muscle. And the conversion of DNA to RNA to protein is regarded as the central dogma of biology. It's that pathway of flow of information is called the central dogma. And if you think about in terms of cost of investigating the sequence of DNA, so what the letters are that make up the DNA, your genetic code, it used to cost, it did cost over $1 billion to sequence somebody's DNA, the first human genome sequence around the year 2000. Now, 20 years later, we're on the verge of it being possible for $1,000.
Right.
And this is just sequencing the DNA itself. And back in the year 2000, it wasn't possible to sequence the RNA. That technology has not really become available till mid to late 2000s, and then it's had a whole upgrade and a step up in its computational power. And now we are now available to do RNA sequencing with service providers. The most commonly used one is Illumina, but there are other companies like BGI that have the ability to do RNA sequencing. And so the idea is the genetic code, we can sequence and see that. When that gets converted into RNA, we want to read that sequence as well. And the reason why that sequence is important is because that is a sequence that could get converted into protein. And so if you want to know the proteins of the cell, the things that characterize the cell's behavior, you want to know what RNA molecules are being expressed from that DNA. And so that is what RNA sequencing is. And in terms of steroids, that becomes very interesting because when an anabolic steroid binds to its receptor, the androgen receptor inside muscle, it has the ability to bind directly to the DNA. It will cause certain genes to be expressed. They will then create certain RNA molecules that will then get converted into proteins. And there have been studies in muscle fiber cell culture and also in humans where people have been given replacement dosages of testosterone, where we can see what genes are expressed when someone is given a replacement amount of anabolic steroids in cell culture or in humans. And that has been conducted. But as far as I'm aware, there's not been research that's investigated when someone is using super physiological levels of anabolic steroids currently or in the past. What does the RNA molecules look like? What genes are being expressed from this steroid receptor, the androgen receptor, binding directly to the DNA? And we have the ability to preserve from when someone is subjected to a muscle biopsy, both the DNA, the RNA, and the protein, so that we can look at that whole pathway of expression from the genetic material inside their muscle to what genes are being expressed with the RNA to then what proteins are being made. And the theory would be if someone is taking anabolic steroids, then the pathways of growth would be way overexpressed. So you would expect the RNA molecules that are responsible for making proteins like myosin and actin, they should be expressed at a much higher level when someone is taking anabolic steroids and their receptor is binding to their DNA at super physiological levels compared to when someone is not. And the question remains, if you are currently taking steroids, that's the assumption of the picture you should see if you analyze the material. The question is then, if you stop taking anabolic steroids, what happens? Do those RNA transcripts, are they down regulated and their expression goes down? You'd expect that to be the case because the receptor binding has gone down, but there may well be a memory in terms of where the receptor has bound to on the DNA. Because when the receptor binds to the DNA, it will open up certain regions of the DNA, and it's possible for those regions to potentially remain open and not close fully. And so there might be the potential for the expression to be at a high level and maintained even when the steroid receptor is no longer actually saturated with super physiological levels from the cycle the person is currently using. Right. And when we're talking about regions of the DNA being opened and closed, that's a epigenetic memory. And epigenetics is the study of, essentially is not the study of the genetic code itself, so the letters of the DNA, it's the study of how those letters are packaged and opened or closed. And the theory is that things you do in your environment, smoking, being obese, resistance training, potentially taking steroids, put a question mark on that one because that's not been shown, but it has been shown for those other things, that regions of your DNA are packaged and opened and closed differently if you are of a certain obesity level, or if you are smoking, and if you are resistance training. And last year, Robert Seaborn published a fantastic piece of research in Nature. This was his PhD research where he actually exposed people to a single resistance training session and then took a muscle biopsy of their leg and he looked at their DNA, and he found that regions of their DNA had become opened that are responsible for making muscle proteins after just one resistance training session. So that made sense. And then what he then did is subjected them to a resistance training program of multiple weeks. And at the end of that resistance training program, those regions were still remained open and maybe even slightly more open. But then what he forced his participants to do was to no longer resistance train. And the interesting thing is those regions of DNA did not close fully. They still remained slightly open. And then when he subjected his participants back to resistance training, those regions for anabolism opened up even more. And so his title of his paper, the conclusion of his paper, is that muscle exhibits an epigenetic memory of resistance training. So regions of your DNA inside your muscle literally get opened when you lift weights, and those regions that are opened are then more expressed. Then more RNA gets made from those regions, and those regions are responsible for making proteins and causing the enhanced rate of muscle growth and the anabolism that occurs from lifting weights. And even when you stop lifting weights, those regions still remain slightly open. So there is a memory of the previous exposure. And when you go back to lifting weights, those regions can open up even further, and you can get back to anabolism even quicker than if you were to have not done it in the past. So muscle has been shown to have this epigenetic memory of resistance training, and that's not even directly receptor binding to the DNA. That's just a mechanical tension being caused across the muscle fibers, causing a molecular cascade that then causes the DNA to then make these RNA molecules that get converted into protein, causing anabolism. So it's a bit more of an even complicated pathway compared to the androgen receptor binding directly to the DNA and then causing upregulation in RNA transcripts and upregulation in protein synthesis like steroids do. So we would expect that there is potentially going to be a memory from androgen receptor binding superphysiological levels. So RNA sequencing has the ability for us to look at the RNA molecules, and DNA sequencing has the ability for us to look at the genetic code of the DNA letters, and you can then take it a further step forward and conduct sequencing that makes you look specifically at regions of the DNA that are opened or closed, and use something called ChIP-seq for that. It's just a scientific term, but essentially it's looking at regions of DNA that are opened or closed. So that's RNA sequencing, then DNA and other sequencing methods.
Wow. Yeah. No, that's pretty awesome. So when you're looking into that, Alex, is a quite a bit of literature out there, or not so much at the minute, in regards to what you're looking at for that muscle memory type of look?
Yes. So the epigenetic memory is basically an hypothesis. It's not been proven, but we would assume because the receptor is binding directly to the DNA, the regions of the DNA might remain opened or closed because of that previous exposure. The only other indirect piece of evidence we have to give weight to that hypothesis is that a piece of research was conducted now pretty much 15 years ago now, and they biopsied past steroid users, and this was published in 2006. And the guys that they were biopsying had not taken steroids for 1 year, and they had taken them on average for more than 5 years. But they'd been steroid free for at least 1 year. And they found that when they looked inside the proteins inside the muscle, that you can count with technology around 2,300 proteins inside muscle. And of those, around 80 were different if someone had taken steroids compared to someone that had not. And that would mean that when someone is exposed to superphysiological levels of receptor binding in their DNA, that is creating transcripts of RNA, it's creating RNA molecules that are then getting converted into protein molecules that are still lingering on 10 years later. So that would give weight to the idea that the region of the DNA that was opened up from the steroid exposure has remained open because those RNA molecules and those proteins are still being made and still present and different 10 years, greater than 10 years down the line in some instances in some of the people that they biopsied. So that's the epigenetic memory and the hypothesis behind that. But that's only one aspect of muscle memory. The other aspect of muscle memory is the idea of how muscle growth occurs in the first place is that when you train or when you take anabolic steroids, you activate the stem cells inside your muscle fibers called satellite cells. They're called satellite cells because, like a satellite, they're on the periphery, like orbiting the Earth. They're on the periphery, whereas in the muscle, they're on the periphery of the fiber. They're not actually inside the fiber; they're on the outside. The idea is if you train, and also independently, anabolic steroids will also do this. They'll activate those satellite cells, and they will cause the satellite cell to divide in two. Right. One cell will remain as a stem cell, so you don't deplete the amount of stem cells you have inside your muscle fibers. The other one will donate its nucleus into the muscle fiber. And there is a whole sequential pathway of a satellite cell nucleus turning into a myonuclei or a true muscle nucleus, which you call a myonucleus, and myonuclei for plural. That is the structural unit itself that is responsible for making proteins, because that is what contains the DNA. So that's where the receptor binding has to happen to make the RNA to then make the protein. So the more actual myonuclei have inside a muscle fiber, the greater the propensity it has for building muscle. You could think about it in terms of a factory. If a factory only has 2 lines available to make products, the amount of products it's going to make per hour is going to be much less than a factory that has 10, 15, 20, 30 plus lines to make product. And this is the same thing. A muscle fiber that has many myonuclei per unit time has got the ability to make way more proteins than a muscle fiber that has few myonuclei. And some research was conducted now in the early sort of 2011, 2012 kind of time. In mice out of a lab. The head researcher is called Christian Gunderson, and he's written a fantastic research paper about a conclusion and the summary of his work of about a decade now about muscle memory in terms of myonuclei accumulation and retention. So if you were to Google Christian Gunderson myonuclei or Christian Gunderson muscle memory review paper, you'd find this paper. It's written pretty much for the lay public. It's not too complicated. But he essentially summarizes over a decade of his lab's research, and the conclusion from their perspective in mice is when you expose a mice to a resistance training stimulus, it will accumulate myonuclei. But then when you then expose the mice to an atrophying stimulus, so a stimulus that causes muscle loss, although you might lose muscle mass, the myonuclei that have accumulated previously remain, and then they then result in an enhanced rate of muscle growth in the future. And that is the pathway in their mind for muscle memory. And this makes sense because if you were to resistance train and you accumulate myonuclei in your leg, you break your leg, it goes into a cast, you will lose muscle mass because you're sedentary and there's no muscle contraction going on. So protein breakdown will be outweighed by the lack of any stimulation of protein synthesis due to the sedentary nature of the cast. But then when you go back to resistance training, the myonuclei that you've banked from previous resistance training exposure are already there and present. You just have to stimulate them to make proteins with the adequate nutritional environment, and you should be able to rebuild the muscle that you once had. And this is a fantastic concept. And this now extends to public health, because the idea should be, well, everyone should lift weights when they're young. They should accumulate myonuclei and build muscle when they're young, when their testosterone is high, when the number of stem cells that they have inside their muscle is high, because we know in both sexes, stem cell content will decrease as you get older. And in males, your testosterone is decreasing when you get older. So you're losing that potential for anabolism as age passes on. So you should train hard when you're young, build muscle, get in that myonuclei. So when you're older, you don't lose muscle mass, become frail, fall over and fracture your hip, become a burden to public health services. So if we could encourage weight training as a public health message, we might be able to maintain people's muscle mass as they get older. And that's all well and good, but this also then extends to anabolic steroids, because anabolic steroids cause muscle growth by increasing the number of nuclei inside muscle fibers. And Christian Gunderson, his lab directly tested this, and Ingrid Egner is the lead author on the research paper that was conducted. And essentially they gave anabolic steroids to mice for 2 weeks. And as expected, at the end of the 2 weeks, the mice who were exposed to anabolic steroids had much bigger muscle fibers, and they contained much more myonuclei. In fact, their myonuclei levels were about 30% higher. And they then left them inside their cages where they did absolutely nothing for 10 weeks. And at the end of that 10-week period, their muscle fibers had shrank down to the exact same size as the control group. So the group that had bigger muscle fibers because of steroids had lost all of that fiber size. Their muscles had shrunken down to the exact same size. Interestingly, and the key point was, even at this time point, 10 weeks later, which is 12% of a mice's lifespan, the minuclei that had previously accumulated were still elevated, and they were still 28% higher.
Right.
And at this time point, they then exposed the mice to 2 weeks of resistance training stimulus. Put an asterisk next to that because I'll explain that further.
Right.
But for now, we can call it a resistance training stimulus. And after 1 week, the mice that were previously exposed to steroids had a 31% increase in muscle fiber size, whereas the natural mice only had a 6% increase in muscle fiber size.
That's right.
And at the end of the 2-week period, even though the mice that were previously exposed to even though the mice, that were previously natural, not exposed to anabolic steroids, their muscle growth did increase, but it never caught up to the same level as the group that was previously exposed to anabolic steroids. And at the end of the 2 weeks of that training stimulus, their muscle fibers were still 20% larger than the mice that were previously natural. So the conclusion of that paper is the minuclei that accumulate from steroid usage do not go down through time in mice, and they in fact actually cause an enhanced rate of protein synthesis and enhanced rate of growth during a training period after that time point. So if you were to carry the conclusion on into humans, it would be if someone is caught taking anabolic steroids in drug-tested sport, where the current ban is 4 years, you might have to conclude that they're retaining a benefit longer than that time period. Although that's a very difficult thing to try and answer, that is a generic statement, could be said. However, the carrying it over into humans is potentially very difficult to say as a direct carryover. The main problem of having that direct carryover is the resistance training stimulus, where I said we have to asterisk in these animal studies, is it's not really any way replicatable for a human to do. And that is because to cause muscle growth in animals, it's very common for a technique called synergist ablation to be used, which is a fancy way of saying cutting one muscle off the mice's leg so it's forced to walk around on another muscle. So you cut one of the calf muscles off from the mice, so for it to even just walk around and function, it has to bear the weight of its entire body on another muscle that previously was obviously not exposed to the full weight of the mice. So that is a massive increase in terms of resistance stimulus, and that results in a large amount of muscle growth, but it's not any way replicable for a human, because it's a training stimulus that lasts 24/7, and the stress that it induces and the damage it induces will be very difficult for a human to create without surgery, which is never going to happen. So what's now put as a big question mark in these studies that Christian Gunderson has done and his lab, and his wonderful model of minuclei accumulate from resistance training, are maintained and do not decrease through time, is that each time they've caused minuclei to accumulate, they've been doing it via this technique. And that's all well and good as a sort of experimental model, and it's been shown that works in an experimental model, but I mean, is that the case in humans? And so the field in general with muscle memory and minuclei accumulation and retention is now moving towards trying to do human studies. And there is another world-leading lab led by John McCarthy, University of Kentucky in America. They conduct similar research to this lab in Sweden, where they are trying to investigate the role of nuclei in terms of muscle growth. Now, they've interestingly shown that in mice, where instead of causing them to be overloaded by cutting one of their muscles off and out, they actually made the mice run inside a treadmill, and each week they added weight to the treadmill. So it's progressive overload. They started on 1 gram, go up to 8 grams over an 8-week period. It's got the hallmarks of a good program to induce hypertrophy. And as expected, the muscles got bigger in terms of fiber size, and they accumulated myonuclei. But then when they stopped them training and left them in their cages doing nothing and looked back into those same muscle fibers, the myonuclei had gone down. So they have accumulated myonuclei from resistance training, but they weren't maintained through time. And this was an actual more physiological, realistic way of training the mice.
Right.
And so that puts a big question mark to the whole model, because that means when the training stimulus is a bit more realistic, the myonuclei actually might go down through time.
Yeah.
Okay. So that's been published. That's been published about 6 months ago now. That's been out there. Now, this Swedish group has really put their money where their mouth is, because they've actually tried to do a human study. And we have to give credit to them to do this, because it's unbelievably large amount of time and resources that would have been required for this, because they decided that they'd get people into the lab, they'd take a muscle biopsy at baseline, they then would train them for 10 weeks.
Right.
They then would take another muscle biopsy to see how big their fibers got, how many nuclei they've accumulated, and then they left them doing no training for 20 weeks, and then they wanted to retrain them 5 weeks after that 20-week period to then see if that they retained a benefit from the 10 weeks of resistance training that they did 20 weeks before.
Gosh,
so
that's a long time.
A really long time. So training them for 10 weeks, them doing nothing for 20 weeks is 30 weeks, and then add in another 5 weeks of training, 35 weeks, 3/4 of a year. That's pretty impressive, right?
Yeah.
And this is where the story, unfortunately, at the moment ends, because this was only published literally a month ago now. And unfortunately, the conclusion of the paper was that they trained 19 people. It was approximately half men, half women. And of those 19 people, only 7 of them actually accumulated myonuclei after 10 weeks of training.
Right.
And the thing is, they wouldn't have known that at the time. They would have trained these people for 10 weeks, taken the muscle biopsy, stored it in the freezer, detrained them for 20, and then retrained them for 5, and then looked at everything.
Yeah.
But unfortunately, what had happened is they trained these 19 people, put all this effort in, and only 7 of them accumulated myonuclei from the resistance training protocol.
Wow.
So their conclusion was they thought they were doing a progressive they did progressive overload, they did blood flow restriction training, they did what they thought in their mind was the best stimulus to cause myonuclei to be accumulated based on the scientific literature, but still only 7 people accumulated myonuclei from the resistance training. So they say we can't draw any general conclusions from this study because we actually didn't train the people hard enough, or at least they didn't accumulate myonuclei. And so if the whole point of the research is train them, see if they accumulate myonuclei, and see if those myonuclei are retained 20 weeks later, I mean, you just can't answer that question if only 7 of the people out of 19 accumulated myonuclei in the first place. So they put a bit of an asterisk and say, well, this is what we've done, but unfortunately this happened. We can learn from this, and means that any human studies that are coming forward from this have either got to train them for even longer than 10 weeks or even harder, or do some new techniques, or something extra has got to be done. Now, this is where sort of the good of science comes in, because they made all their data publicly available online. And although they're in conflict with this lab in Kentucky, where Kentucky is shown in their mice model that when you train a mice on a treadmill, the nuclei that accumulate from it go down. And whereas in Sweden, they're saying the nuclei that accumulate from mice are retained forever. So they're a bit in a conflict in the scientific literature, but because they've made their data publicly available, anyone can scrutinize it. And a researcher called Kevin Murak out of this lab in Kentucky has looked at their data. And when you look at those 7 people, and Kevin Murak did this analysis and published it about a month after that Swedish study was published. When you look at those 7 people that have accumulated myonuclei, all 7 of those people, after the 20 weeks of detraining, lose the myonuclei. And the graph is incredibly clear. At baseline, they have a low level of myonuclei. After 10 weeks of training, in all of these 7 people, it's significantly higher. And then after 20 weeks of doing nothing, the numbers have dropped down back to baseline. And so this means in humans, the myonuclei that have accumulated from resistance training are not being retained through time, at least in this subset of 7 people.
Yeah.
And this is where it gets difficult, is because this is only 7 people, and we're looking at in a subset of category of people where out of those 19, the majority of those people were not accumulating mononuclei anyway in the first place. And you're looking at the subset that did. And so the question now comes into, well, what really causes a myonucleus to have permanency? What causes it to be retained forever? And now what the researchers are starting to question is, well, it might not be as cut and dry as they thought with the mice studies, that once a stem cell gets activated, splits in two, and that nucleus of one of those cells then starts to migrate into the muscle and become a true muscle nucleus, that there is a massive molecular cascade where there is a transition of identity of becoming a stem cell into a muscle nucleus proper. And that time course has been studied extensively, and there is a whole molecular cascade where markers on the outside of that nucleus change from being what was regarded as a stem cell to then what is regarded as a true muscle nucleus. And there is a bit of a time course. And so the question now is becoming, maybe a nucleus could go inside the muscle fiber and it still not have gone all the way to 100% being a true muscle nucleus, still could potentially maintain some stem cell identity, and that